Hello students of Standard 10. Welcome to the e-learning classes of DAV Moyle Public School. Subject Science. Our today's topic is how do organisms reproduce? Before we discuss the mechanism by which organisms reproduce, let us ask more basic questions. Why do organisms reproduce? After all, reproduction is not necessary to maintain the life of an individual organism. Unlike the essential life process such as nutrition, respiration, or excretion. On the other hand, if an individual organism is respiration or excretion going to create more individuals, a lot of its energy will be spent in this process. So why should an individual organism waste energy on a process it does not need to stay alive? It would be interesting to discuss the possible answers in this topic. Whatever the answer to this question, it is obvious that we notice organisms because they reproduce. If there were to be only one reproducing number of a particular kind, it is doubtful that we would have noticed its existence. It is the large number of organisms belonging to a single species that brings them to our notice. How do we know that two different individual organisms belong to same species? Usually we say this because they look similar to each other, thus reproducing organisms create new individuals that look very much like themselves. Do organisms create exact copies of themselves? Organisms looking similar because their body designs are similar. If body designs are to be similar, the blueprint of these designs should be similar. Thus, reproduction at its most basic level will involve making copies of the blueprints of the body. In the previous class, we have learned that the chromosomes in the nucleus of a cell contains information for inheritance of features from parents to next generation in the form of DNA, that is deoxyribonucleic acid molecules. The DNA in the cell nucleus is the information source for making proteins. If the formation is changed, different proteins will be made. Different proteins will eventually lead to alterations or altered body design. Therefore, a basic event in reproduction is the creation of DNA copy. Cells use chemical reactions to build copies of their DNA. This creates two copies of the DNA in a reproducing cell, and they will need to be separate from each other. However, keeping one copy of DNA in the original cell and simply pushing the other one out would work because the copy pushed out would not have any organized cellular structure for maintaining life processes. Therefore, DNA copying is accomplished by the created or creation of an additional cellular apparatus, and then the DNA copies separate, each with its two cellular apparatus effectively a cell division to give rise of two cells. These two cells are of course similar, but are they likely to be absolute identical? The answer to this question will depend on how accurate the copying reaction involved occurs. No biochemical reaction is absolute reliable. Therefore, it is not only be expected that the process of copying the DNA will have same variations each time. As a result, the DNA copy generated will be similar, but they may not be identical to the original. Some of the variations might be so drastic that the new DNA copy cannot work with the cellular apparatus it inherits. Such a newborn cell will simply die. On the other hand, there could still be many other cell variations in the DNA copies that could not lead to such a drastic outcomes. Thus, the surviving cells are similar to, but subtly different outcomes from each other. This inbuilt tendency for variations during reproduction is the basis of evolution, as we will discuss it. The importance of variations. Population of organisms fill well-defined places or niches in the ecosystem. Using their ability to reproduce, the consistency of DNA copying during reproduction is important for the maintenance of body. Design features that allow the organisms to use that particular niche. Reproduction is therefore linked to the stability of populations of species. However, niches can change because of reasons beyond the control of the organisms. Temperatures on Earth can grow up or down, water levels can vary, or there could be meteorite hints. To think of a few examples, if a population of reproducing organisms were suited to a particular niche, and if the niche were drastically altered, the population could be wiped out. However, if some variations were to be present in a few individuals, if these populations, there would be some change to them or for them to survive. Thus, if there were a population of bacteria living in temperate waters, and if water temperature were to be increased by global warming, most of these bacteria would die, but the few variants resist to heat would survive and grow further. Variations is thus useful for the survival of species over time. Modes of reproduction used by single organisms. First one is fission. For unicellular organisms, cell division or fission leads to the creation of new individuals. Many different patterns of fission have been observed. Many bacteria and protozoa simply split into two equal wholes during cell division. In organisms such as amoeba, the splitting of the two cells during division takes place in any plane. However, some unicellular organisms show somewhat more organizations of their bodies, such as in Leishmania, which have a white-like structure at one end of the cell. In such organisms, binary fission occurs in a definite orientation in relation to these structures. Other single-celled organisms, such as the malarial parasite Plasmodium, divide into many daughter cells simultaneously by multiple fission. Yeast, on the other hand, can put out small buds that separate and grow further. Next one is fragmentation. In multiple cellular organisms with relatively simple body organization, simple reproductive method can still work. Spirogera, for example, simply breaks up into small pieces upon maturation. These pieces or fragments grow into new individuals. Can we work out the reason for this based on this is not true for all multicellular organisms. They cannot simply divide cell by cell. The reason is that many multicellular organisms, as we have seen, are not simply a random collection of cells. Specialized cells are organized as tissue, and tissue are organized into organs, which then have to be placed at definite positions in the body. In such a careful organized situation, cell by cell division would be impartial. Multicellular organisms therefore need to use more complex ways of reproduction. A basic strategy used in multicellular organisms is that different cell types perform different specialized functions. Following this general pattern, reproduction in such organisms is also the function of specific cell type. How is reproduction to be achieved for a single cell type? If the organisms itself consist of many cell types, the answer is that there must be a single cell type in the organisms that is capable of growing, proliferating, and making the other cell types under the right circumstances. Next one is regeneration. Many fully differentiated organisms have the ability to give rise to new individual organisms from their body parts. That is, if the individual is somewhat cut or broken up into many species, many of these pieces grow into separate individuals. For example, simple animals like Hadrilla and Planaria can be cut into any number of pieces and each piece grow into a complete organism. This is known as regeneration. Regeneration is carried out by specialized cells. These cells proliferate and make large number of cells. From this mass of cell, different cell undergoes change to become various cell types and tissues. This change takes place in an organized sequence referred to as development. However, regeneration is not the same as reproduction, since most organisms would not normally depend on being cut upon up to be able to reproduce. Next one is budding. Organisms such as Hydra use regenerative cell for reproduction. In the process of budding, in Hydra, a bud develops as an outgrowth due to repeated cell division at one specific site. This bud develops into tiny individuals and then fully mature, detach from the parent body and become new individual. Vegetative propagation. There are many plants in which parts like roots, stem and leaves develop into new plants under appropriate conditions. Unlike in most animals, plant can indeed use such as a mode for reproduction. This property of vegetative propagation is used in methods such as layering or grafting to grow many plants like sugarcane, rose or grapes for agricultural purpose. Plants raised by vegetative propagation can bear flowers and fruits earlier than those produced from seeds. Such methods also make possible the propagation of plants such as banana, orange, rose, and jasmine that have lost the capacity to reproduce seed. Another advantage of vegetative propagation is that all plant produce are genetically similar enough to be the parent plant to have all its characteristics. Similarly, birds produce in the notches along the leaf margin of bryophyllum falls on the soil and develop into new plants. And the last one is spore formation. Even in many cells, simple multicellular organisms, specific reproductive parts can be identified. The thread-like structure that develops on the braid above are the hyphae of the braid mound, that is, rhizophus. They are not reproductive parts. On the other hand, the tiny bulb on a stick structure are involved in reproduction. The bulbs are sporangia, which contains cells or spores that can eventually develop into new rhizophus individuals. The spores are covered by thick walls that protect them until they come into contact with another moist surface and can begin to grow. All the modes of reproduction that we have discussed are so far allows new generation to be created from a single individual. This is known as asexual reproduction. Now it's time to check your knowledge about today's topic. Here are some questions as your assignment. I hope you understand today's topic. We again meet with new video. Till then, stay at home, be safe, be happy.